Are you doing an intro or am I just going?
No, we already know who you are.
Okay, very good.
Gentlemen, Alan Booker.
Yeah. Well, this time what we're going to do is, I was trying to think of how we're going to do this because we've got an hour and then we're going to break for lunch and then we're going to have another hour. And so I got here and we realized we had 4 microscopes. Usually I just have one and we just demo up front, but it's like we might actually be able to do some hands-on this time. So what we're going to do is towards the end of this hour, we're going to spend a few minutes getting the microscope set up.
Your phones and Jake, shut up.
Yeah, we're going to actually spend like the last 15 minutes of this session kind of getting the microscope set up and going over a few things. And then when we come, then during lunch, we'll get our samples, we'll bring them back. That way we'll have an entire hour to do some microscopy together. But we'll start off by preparing a slide here, looking at it together. I've got it set up where I should hopefully be able to take what I'm seeing on this microscope and put it up on the screen. And that way we can kind of look at it together. And then we can break up into groups and we can prepare slides. I think what we're going to end up doing, because of equipment, is we're going to take that table back there and we're going to set it up as our slide prep location. So when you want to prepare a slide, you'll bring your sample there. We'll have everything there for you to prepare the slide. Then you'll take it to your microscope and look at it there, because I don't think we have enough stuff to I haven't looked at it, but I don't think we have enough stuff to have slide prep going on in 4 locations at one time, right? So probably what we'll do. I need to find out what everybody else has with their microscopes. We'll find that out in a few minutes. But we will need, I do have several of these, but we'll need these. This will be where you'll be mixing everything. We'll need a number of pipettes. And so one of the things I will need is 2 little cups of clean, like spring water that don't have chlorine. One is for, well, we'll get to that later, but we'll need 2. We'll need 2 clean water sources, two little cups with water in them before we're done here, okay? And that'll need to be at the slide prep station, okay? All right, so what I want to do first is, this is a refresher because I know everybody was here last year, right? Okay, and so we went through this presentation last time and it has a number of pictures that I wanted you to see. But so this time, since I taught it last year, right, then what are we going to do this time? You're going to teach it this year, okay? So here we go. We're going to do this fast. This is sort of a review. Everybody remember this guy? Who is this? Yeah, it's Fukuoka. So let's see, who wants to read it? Go right ahead, yeah.
Many people think that when we practice agriculture, nature is helping us in our efforts to grow food. This is an exclusively human-centered viewpoint. We should instead realize that we are receiving that which nature decides to give us. A farmer does not grow something in the sense that he or she creates it. That human is only a small part of the whole process by which nature expresses its being. The farmer has very little influence over that process, other than being there and doing his or her small part.
Right. So in other words, how do we partner up with the natural ecosystem to grow nutrient-dense real food? Right, and that's what we're going to talk about. This difference, which is the difference between ecological and chemical cultivation, right? So who here wants to tell me what their memory is of what is chemical cultivation? And don't just read it off the screen. Like when I say chemical cultivation, what are we doing?
Spraying fertilizers.
Yeah, and what are those fertilizers typically? What are they looking like? What are they chemically like? NPK, but what are those chemically? Who are my chemists?
Inorganic.
They're chemical salts, right? And what is a chemical salt? Something that dissociates in water. Because what's happening here is we're basically putting a chemical down that has nitrogen, potassium, phosphorus. Those are the 3 biggies, right? We're putting those down in a way something that dissolves into water, and then when the plant drinks, it has to take that in, right? And is NPK what the plant needs? All of it? No. So what is ecological cultivation? Why is it fundamentally different? Somebody give me that.
you're using basic healthy soil to make available whatever the plant needs to take out of that.
Exactly. And what's the difference between soil and dirt?
Well, we discussed this. Soil usually is composed of clay and sand and silt is microorganisms that are alive. Dirt is inorganic and has nothing living in it.
Yeah, so dirt is just the sand, silt, or clay, sometimes called mineral soil, but we'll just call it dirt. Okay, it's not a living thing. It's not a living ecosystem. Soil is a living ecosystem, and so one of the ways that we put it is we feed the soil and the soil feeds the plant. We work with and we get the soil into really great shape. So one of the reasons that we would take a microscope and look at soil is because if we're doing ecological cultivation, which is what it was required to get nutrient-dense food, then what we're interested in is the entire microbial workforce there. You know, do we have all the different types of workers we need? Do we have enough workers? And are they all doing their thing? And if they are, then we're going to be in much better shape to get ecological cultivation happening, right? So that's part of the reason we're pulling the microscopes out is we're looking. It's like the factory floor. We go out and we look and see if all of our workers are there, and all of our specialists are in their place and doing their thing. Because if we're missing half our workforce, then probably we're not going to have the factory working very well, right? Okay. So, yep, soil composition. We're going through this super fast. Soil is inorganic matter. That's the sand, silt, or clay. Yep. Organic matter. Okay, that's SOM, soil organic matter. Soil life. These are bacteria, fungi. I should add on to that archaea, protozoa, nematodes, etc. Water dissolved gases, and Here's the little thing to recognize, and it's not pointed out very often. If you have soil which is closer to dirt that has very low soil organic matter, it's really hard for the soil life to get a foothold in the very beginning. It doesn't have the conditions it needs to thrive. There's a magic threshold, and in practice it seems to be somewhere around 2.5 to 3% soil organic matter. Below that, you're really trying to get the soil happy and stay in there and work. It's because you don't have the structure in place for your workforce to be like, we like it here. We have everything we need to thrive. Once you get up to about 2.5 to 3%, then it's like, yeah, this is better, right? We're happier. And one of the things, if you don't have that 2.5 to 3% and you want to get yourself started, you need to bring in some extra condominiums and some extra soil, cation exchange capacity. What's one thing you might be able to add, Opalyn? Biochar. And Jack, right? It's like, this is the reason that Jack and Opalyn both have been like, biochar. It's like, yeah, it actually can help be one of the elements that helps you bridge from dirt to soil by giving you structure, water retention capacity, cation exchange capacity, housing for your workforce, all of that. And it's one of those things. So it's one of the reasons you've heard a lot about biochar recently is if you're trying to do ecological cultivation and you don't have the soil organic matter yet, right, or you have other structural problems with your soil, then there are reasons that biochar, this is part of the reason that it was used widely in the Amazon, because in the tropics, soils are typically very thin and very nutrient deficient for ecological reasons we don't have time to get into today. Okay? All right. So sand, silt, and clay. I just put this in here last time to show you the difference in size. Okay? Yeah, that big thing is the average size of a sand particle. Silt is that, look how small clay is compared to that. Okay, and there's a reason that clay is what will help make something waterproof, is it's little tiny and it can stack on top of itself and compress to the point where it's like shingles and it gets compressed in there. There's no space for water to get through, and that's why it's waterproof. That's the inorganic part of our soils, right? And then remember this thing, I don't have time to go through all of it, but this is when you have sand, silt, and clay in different percentages, what do you have? Do you have clay loam or sandy clay loam or whatever, right? In the ideal world, we'd be somewhere in that loam or clay loam area, but in reality, you get what you get. Maybe it's heavy in clay, or it's heavy in silt, or it's heavy in sand. And anyone that's super heavy in one of those directions, all by itself has problems. When we start to get the soil life going, then it will help structure this, create soil organic matter. And here's the magic. If you happen to be in one of these imbalanced edges and you can get soil organic matter going, it'll help make it look like that nice loam in the middle. It will kind of help you fake having nice loam, okay? And biochar can help you move in this direction in many cases, okay? So it's one of the reasons we look at, it's like, hey, I got really heavy clay. What do? Well, I could add some biochar and actually be helpful. What if I have sand? I can add some biochar and it'll be helpful. It's like, so that's one of the reasons that we've looked at different ways of making biochar and so forth. All right. Chemical composition of plants. I don't have time to break this down an awful lot. Carbon, oxygen, and hydrogen are actually 96% of the plants. If you remember last time we talked about how it is that the carbon and the oxygen actually come from the atmosphere as the byproduct of CO2 being metabolized, right? So that it's actually more true that plants, when somebody says, oh, that plant is growing up out of the ground, you say, oh, no, it's not. It's precipitating out of the air, which is actually, from a mass perspective, much more technically accurate, that plants precipitate out of the air than they grow up out of the soil. How's that? That's great to share with kids or high school students or whatever. It blows their mind and opens up all kinds of great conversations, right? Okay, but when we get down into those things, we get into the macronutrients, right? The nitrogen, phosphorus, potassium, calcium, magnesium, sulfur. And in chemical cultivation, what we typically see is that the NP and the K, the nitrogen, phosphorus, and potassium, are thrown down there as chemical salts. As soon as water hits it, those chemical salts dissociate. That is, that anion and the cation go like this. And it's those cations that are the things that go up into the plant in the water. The problem is that the plant can't really regulate that very well, because it's like if we throw a lot down there and the plant needs to drink and there's way too much nitrogen dissolved in the water, for example, what can the plant do about that?
No choice.
No choice. This is why you can burn a plant with nitrogen fertilizer. It's because it has to drink to stay alive. And if you've put down all this stuff that just dissolves in the water and it has to drink the water, it can take in way too much nitrogen, more than it wants. One of the advantages of ecological cultivation is that the plant gets to choose a lot more specifically what it wants. And it can signal down to the workforce in the soil saying, hey, here's what I want, and here's how much I want, and when I want it. And this is what gets really fun about it. That particular root hair can signal completely differently than that root hair over there, because this root hair is feeding that leaf that's over here and getting more sunlight, and this root hair is feeding up to that leaf that's over here more in the shade. And at that moment, they need very different things. Okay? And so when we're doing ecological cultivation, the plant can actually selectively ask for what it needs in different zones of the plant. On different branches, can ask for different things in different moments. Fun, huh? All right. And then, of course, we have the micronutrients: boron, chlorine, copper, iron, manganese, zinc, molybdenum, nickel, sodium, and there's more. But these are some of the principal micronutrients. And chemical agriculture basically ignores these and pretends they don't exist, mostly. Okay? And as a result, you never actually get hyper healthy, nutrient dense plants out of chemical cultivation. Okay? Remember von Liebig's law of the minimum. So somebody explain von Liebig to me. Put it into common language. What is von Liebig's law of the minimum?
plants will grow as best they can up to the least amount of the mineral that's missing.
Right. Like if you were trying to, yeah, what do you
I
want?
was going to say, a team's only as strong as its weakest member.
Right.
Yeah, that's better.
How about this? If I say, here are a bunch of components to build computers, build as many computers as possible.
And we have 300 monitors and 20 keyboards and 3 disk drives. How many computers can we build?
Right? We can build 3, right? So this is von Liebig's law of the minimum. The number of computers we could build are limited by the most limited resource, right? If it's a critical, and the same thing is true in living systems. Like for example, in some living systems, the total amount of biomass that can be produced is limited by phosphorus availability, right? It's like, yep, we don't have more phosphorus to create more biomass, so we're just done in this particular place at this particular moment. We can't produce because phosphorus is not, there's an idea in economics called substitutionality, right? Right, John? Substitutionality. And substitutionality is the idea that, hey, if this product is we can't get it, we can substitute another product. Well, in life, substitutionality doesn't exist like that. Phosphorus is phosphorus. You must have phosphorus. You can't say, well, we don't have enough phosphorus, so we're going to use potassium instead. Does not work that way, okay? So you have to have it. So living systems like this are limited by von Liebig's law of the minimum. And so what we're doing when we are doing ecological cultivation is we're trying to set things up so that the ecology can provide all of the nutrients required at the required level at the required time so that the living system can be as abundant and healthy as possible. That's literally what we're up to. Okay? So, how do plants uptake nutrients? There's really 2 major pathways. And what's interesting is that I have shown this slide in classrooms, like graduate level agronomy classrooms. And they looked at me like, huh? It was really interesting, right? Because what's happening is in a lot of these agronomy classrooms, the whole plant symbiosis with soil microorganisms is ignored. Okay, and the only thing they talk about when they talk about plant nutrient pathways is they talk about these chemical uptake pathways. And they'll talk about how it is that, yes, the roots of plants can exude a mild carbonic acid to basically break loose some of these anions from like clay particles and get them up in the water and get them in. I'm like, well, what about soil microbial life? And they're like, they don't talk about that. Why do they not talk about that? They kill all of it with the pesticides, the herbicides, and the fungicides. So if you are doing industrial agriculture, the reason they don't talk about that second way that plants get their nutrients is because all of the biocides that they are using are basically killing a large majority of that workforce. Okay? They are, they're basically, the way I put it is this when I'm talking to a graduate level agronomy class or a 300, 400 level agronomy class, is what you're doing is you're manufacturing a plant system on a sterile dirt substrate outdoors by applying chemicals in this way and ignoring von Liebig's law of the minimum. And therefore, you will always have unhealthy plants. And they will basically, what will happen is nature will try to fix your problem. And how does nature try to fix your problem?
Bugs.
Exactly. It sends along the workforce to get rid of all of those nasty, very sick plants so something better can grow. So what you've done is you've created plants that are so sick and so unhealthy that nature says, send in the aphids, send in the Japanese beetles, send in the, right, all the things. And so what happens is that they're coming in and trying to get that out of there because in nature, it's like that's not good enough to survive. Okay? So what you've done is you've successfully grown insect food, not human food. Okay? And then you try to keep the insects off of it long enough that you can feed it to humans. And then you wonder why the humans get sick.
And ironically, if you just let it happen, it would all become organic matter and go down to the soil that you're trying to fix. You
Yes.
can't fix because it doesn't have enough organic matter.
Right. So nature's trying to fix it, and you're trying to like unfix what nature is trying to fix because you want it to go your way. You want to like manufacture a plant. You know, it's crazy, but that's what we do. So it's important you understand that yes, the water solubility is there. It's what chemical in agriculture is based upon, but the symbiotic relationship between the soil microbes and the roots of the plants, very important. So there's your chemical cultivation. We kind of talked through that. The reason, by the way, when you get into chemical cultivation, they get so obsessive about pH in soil is because different of these minerals become basically soluble in water in different pHs. And so what happens is you're really dependent upon soil pH. And here's the trick, though. And this is something that we need to understand. When you're first getting started, and your soil isn't healthy, this is still true. Your workforce isn't there. So in the very beginning, yes, I'm concerned about the pH. I want to make certain the pH is decent so that we can get things moving. But here's the interesting thing. Once you get soil organic matter in abundance and you get a really solid soil food web, then those microbes will actually buffer the pH right around the root zone so that they can make the nutrient exchanges that they need back and forth between themselves and the plant roots. So yes, when you are getting started, pH important, and possibly mineral balancing important to get things going. Once you're up and going, then the soil food web will take care of a lot of the problems, except for major issues. But we don't have time to get into all that today. Okay? Cation exchange capacity. Somebody explain to me real quick. Who can do CEC? Cation exchange capacity.
You can.
I can. Oakland's laughing because she's like, I don't know how many times she's seen this slide.
I'm usually not allowed to play, so I'm
Right.
trying really hard not to play.
Yeah,
Guys, come on.
Oakland's one of the instructors on my team, so she's usually not allowed to play when I'm doing this. Right? This is trying to make you think about it a little bit, and it's also really interesting for me to see who in the class is like knows these terms. I don't have time to like dig way into CEC, but what it basically is this. When you have something like a clay particle, it has sharp little edges on it, and those sharp little edges will basically hold electrostatic charges. And that electrostatic charge is a little point where a cation can come in and kind of like hang out there because it likes it. Oh, you're a negative charge and that's a positive charge, and we'll just kind of hang out together, right? And so the whole thing is like sand particles are typically really big and round, and they have a really low cation exchange capacity. That is why when you put water through sand, all of the nutrients just go because the sand can't hold on to it. It doesn't have any electrostatic grip, right? But if you have something like clay, which has got all kinds of sharp little edges on it, electrostatically, if there are cations hanging around, then they'll get electrostatically held on to, and when you get water coming through it, you don't just get all of it flushed through, okay? So this is something important to know. This is what you see here. Here's some examples. Like light-colored sands can have a cation exchange capacity of 3 to 5. It's like nothing, right? Dark-colored sands can go 10 to 20, but by the time you get to organic soils, you're up to like 50 to 100, okay? Now, the other interesting thing is, and this is just the inorganic side of things. As soon as you get soil life in, the soil life goes, oh, wait a minute, right? I'm going to get to exchange all of these nutrients for goodies from the plant a little later. So what do I want to do? Store. So what it will do, the bacteria, the fungi, the and so forth, those will go into the soil and they will grab these nutrients and they will hold on to them, right, in their back pocket until the plant comes along and says, hey, I really need that and I'm paying. What do they pay in? What does the plant pay in? Sugars, starches, other metabolites. Specifically, the thing is, there, it has its solar panels going, and it's got energy, right? And if you're a microbe down in the soil, you're not photosynthesizing, you need energy. So it's like, hey, tell you what, here's the exchange. Jack, you're up there on the upstairs. You've got all kinds of energy, right? I'm down here. I got all the nutrients. I need some energy. You need some nutrients. I'm gonna throw some stuff down in the barter blanket, right? Can we make a deal, right? Right. And we'll sit there and we'll just barter back and forth. And so it's a mutually beneficial exchange. And that's literally how this is going on. Okay.
Sometimes I kill you and take all of
it gets really interesting. It gets really interesting, right? Or sometimes there's stuff goes on like the rhizophagy cycle and so forth. We don't have time to get into today, where certain bacteria will go inside of the plant and certain things happen, and then you get spit back out, and it's like, and the plant takes back up the exudates it put out. It's like it gets really, really interesting, right? But I'm trying to just get us to high level. What we basically need is we need all these workers there in sufficient quantities. And when we get to the microscopes, what we're going to be seeing is, are they there, right? Do we have good guys or we have bad guys, and are there enough of them, right? Because what we're trying to do is get this down to the point where you don't have to have a PhD in biology and soil ecology in order to get done what you want to get done. What you can do is just pull out your microscope and do a quick and dirty check to make certain that you got all the good guys there and active. And if so, they know what they're doing. They know, you got all the workers. They're all expert workers. They know a lot more about the work than you do. So let them do their work, right? Hire the right workers and then let them bring their expertise to the job. That's what a good manager does, right? And what we're trying to do is we're using the microscope to make certain we have the right expert workforce on the job and that they have everything that they need in order to get their job done. And if they don't, then we're going to help change those conditions around. That's how we're looking at this, right? Okay. So I'm going to skip all the stuff on soil testing, chemical soil test. Okay, we talked about this before, ecological cultivation. These are a lot of the organisms, right, that are involved, but we're not going to try looking at everything through all the trophic levels. What we're going to say is that really foundationally in the soil, bacteria and fungi are like the foundational layer. There are a few archaea, but you're not going to be able to tell the difference between the archaea and the bacteria, so just call them bacteria for now. Okay? And then the next sort of up from that, trophically, are the protists. And the ones we're going to see a lot of are we're going to see flagellates, ciliates, and amoeba, and we're going to talk about those in a minute. And then up from that, we're going to see nematodes. And there's a great trivia question for you. In terms of population, what animal on the planet has the largest population? They're not animals. They're in a different domain, remember?
I call them little bitty animals.
They're not animals. So the Animalia is in the domain of eukarya. So these got to be eukaryotes. But animals are all multicellulars. Nematodes are actually the most abundant animal on the planet. Yeah, most of them are good. So, all right. In soil cultivation, generally think about this. Aerobic is good and anaerobic is bad. Okay, and for the scientists in the room, aerobic means above 6 ppm of oxygen or 6 milligrams of O2 per kilogram. Okay? So here's the thing, and I'm just going to throw this out because it's actually important. I've begun from working with people who have been playing with this for a while. A lot of folks who are like doing soil food web school and so forth, they really concentrate on the aerobic organisms. And they talk about them and it's like, and here's the problem. The problem is that in real soils, small zones of your soil will be driven anaerobic periodically by different things. It'll rain and you'll get anaerobic temporarily or whatever. What we've found, and this is from people actually experimenting with it, is that yes, it is really good to have all of the aerobic organisms there and to pay a lot of attention to them, But you get even better results if you get beneficial anaerobes in there as well. And my suspicion, we haven't been able to prove it yet, but this is my suspicion from watching it, is that what happens is that most of the plant pathogens, the things that cause problems, are either facultative anaerobes or anaerobes. And remember competitive exclusion from yesterday? If we get a lot of good guys, anaerobes in there, when anaerobic conditions come up, they're sitting there dormant. They're in sporal stage or whatever, they're going dormant. And all of a sudden we get anaerobic conditions. There could be some spores of bad guys as well. But if we have a lot of spores of the good guy anaerobes, they come alive and they competitively exclude the bad guys from getting a big foothold. That's my hypothesis at the moment from what we've seen. So we actually like to get some good anaerobes in there as well. Okay, I'm just going to leave it at that. That's just some research that we're up to these days. But if you see us actually experimenting with inoculating soil with beneficial anaerobes as well as aerobic organisms, that's part of the reason why. Okay, let's see how I'm doing. All right. Okay, so we're going to use soil microscopy in order to look at all of this. What we typically need is we need a microscope that can go from, well, we're going to use a 10x eyepiece and then objectives from 4x to 40x so that we can get a total magnification of 40 to 400 times. I think I have my 60x objective on this one at the moment. We'll be looking at that. I have a really nice, expensive 60x on this one, but you don't really need that. A lot of the soil microscopes will come with a 4x, a 10x, a 20x, and a 40x objective. Okay, and the way you figure out your total magnification, and by the way, they'll almost always send you like these 20x eyepieces. Don't use those. I won't get into all the optical reasons, but use the 10x eyepiece and then change. So just leave that and then change your total magnification by changing the objective you're looking through, right? So your eyepiece is up here. These little guys down here are called the objectives. They do the heavy lifting of magnifying, and they're the ones that will go from 4x to 40x, okay? So this is technically called a compound microscope, and what we are using is technically called bright field, because what we're going to have is down here at the bottom, we have a light, and it shines the light up through the sample. In other words, when we look down, we're going to have light shining right up in our eyes. That's why it's called bright field. The field around everything is bright, and as opposed to dark field, and there's ways of using a condenser where all the light comes in at an angle, so that when you look down, you don't have a light shining in your eyes. It's illuminated differently. So that's the difference between light field and dark field microscopy. There are kinds of microscopes that are called stereo microscopes, but they work on reflected light, and those are used when you're looking at an opaque object the light can't come through. Okay, so if you're going to look at, say, leaf structure, and the leaf won't transmit light very well, you put it under your stereo microscope and you light it from the top and the light bounces off and you can look at it. So what we're looking at here is a compound microscope. So if you go looking at microscopes online, it says, do you want a stereo microscope or a compound microscope? For soil, we're going to use a compound microscope. Okay?
What do they run?
You can get a reasonable one for just a couple $100. You can go $300, $400 and get a pretty reasonable soil microscope. Okay, you can go way higher than that if you want to do like the ones that we're going to epifluorescence and that kind of stuff that we really should be doing for like high-end work is about $4,000 to $5,000, and you can go up to $10,000 easily if you're doing high-end work. But for what we're trying to do here, you can get by with a $300 to $400 microscope. Okay? All right. So the foundational trophic layers I mentioned earlier, we're going to look at bacteria, archaea, fungi, protozoa, nematodes, and then we're also going to look at organic matter, although it's not really part of the trophic layer. It is the environment, right? So we start seeing things like, oh, we have protozoans, we have, we got little clumps of organic matter and so forth. And this is what we're going to be looking at. This is why it's called bright field. See how it's all nice and white because all the light's coming up through it. And then you can see that the light is coming through these things, and we're looking kind of through them translucently, right? Okay, so good guys versus the bad guys. This is the language I use when I'm teaching high schoolers or whatever. We get into it and teaching advanced scientists. We have to be a little more prim and proper about the language, but for right now, we can just think about good guys and bad guys, okay? So when we use the microscope, we make sure our sample is full of good guys and has very few bad guys. The good guys are all aerobic organisms, and almost all the bad guys are anaerobic organisms, right? And what we have here is spirochetes. What do you think? Good guy or bad guy? Bad guy. These are anaerobes. They're a pathogenic anaerobe. And how do we do that? Just by what we're going to call qualitative analysis. Okay, qualitative. What's its size? What's its shape? What's its color? It's also called morphological analysis. Morph is size and shape. Okay, so we're looking at that and going. You can see why they're called spirochetes, right? They're spirals. Okay, and when we start looking at it, we'll start looking at it and realizing, oh well, okay, in a 400x, 1 micron is about that big. Or order it's in, but we'll be able to get good enough to be able to tell us a lot about what's going on without having to do like genomic analysis. Okay. Now, if you want to do the genomic analysis, go to it. It's actually doable at home now if you have the money to spend for the genomic devices. It's just a few $1,000s. So if I were doing like a large research farm, we would actually have that kind of stuff because it would be worth it. Okay, so in other words, this is the sort of thing you're going to be looking at. So what in the world do we see here? This is a field view from a compound microscope, 400x. We're seeing, this is a root. What do you see here? Num num. Somebody has come chomping on that root. Probably a root-feeding nematode, right? By the way, how many of you, it's like nematodes are bad. How many of you have that idea? Anybody? Because what happens is if you go to the typical garden center and you say the word nematode out loud, what do?
Poison.
Poison it. It's because when you say nematode in the garden center, they think you're talking about the one bad type, which is a root feeder. And they're like, well, what you got to do is you got to poison them. You got to get them all rid of them and so forth. And it's like, no, because the root feeders come out when you have bad soil conditions. You need to fix the soil condition, right? But the problem is that there are actually several types of nematodes that we really want because they're a critical part of our workforce. And so we got to be a little more subtle about this and know the difference between the good nematodes and the bad nematodes. That's telling me that there's probably a bad nematode around somewhere, right? Okay, this is a ciliate. It's called a ciliate because cilia means hair. See the little hairs kind of coming off of it? Okay, we're getting some coccibacteria, these little round things. Okay, some soil organic matter here. And so that's what we're kind of seeing. What do you think? Is this, are you liking this or not liking this?
It's not got very much bacteria in it.
Yeah, it's kind of low in good bacteria. Ciliates are ciliates good or bad? So we need to figure that out, right? We got a root-feeding nematode loose. So this is kind of the questions we're asking when we're looking at a slide, right? So let's go looking at the good guys and bad guys, okay? So the cocces are your round ones. Your bacilles are more of your rod-likes, okay? There's your spirochetes. Your vibrios are like a comma and so forth. And so you can see, like this is the helical forms and so forth. You start realizing that, yeah, like most of the cocces are pretty good. Most of the bacilli are pretty good. However, the vibrios and the spirochetes are typically bad. And so this is the sort of thing we're going to be looking at. So we've taught middle schoolers to do a soil analysis before because it's like, does it look like that or does it look like that? Right? And how big is it? And so forth. And they're able to go, oh yeah, I'm seeing a lot of good ones. I'm not seeing many bad. It's like it's not that complicated once you kind of get down into it. Okay? Well, then we get into fungal hyphae. Okay? This is a good fungal hyphae. The way you're going to tell a good fungal hyphae is that they are typically and that they are segmented. See these little segments right here? It's a little hard to see on the screen. See the little segment? Okay, kind of like bamboo is segmented. Okay, it's going to look like that. If it's not segmented, then it may not be good fungi. It may not, so forth. What we're looking at when we look at a fungus, we're looking at what color is it and how big is it. If it's really small and it's white, it may very well be a bad fungi. Most of the good fungi are going to be 2.5 to 3 microns across or bigger, and they're going to be colored. Okay, so that. How about amoebae? These are one of our 3 types of protists. These are eukaryotes, and these actually feed, these and the others feed on the bacteria, and they help keep the nutrient cycling going. They keep, because otherwise you can get a bunch of one type of bacteria, they're overgrowing, and they all grab a hold of all the nutrients, and then when you have these guys, it's kind of like the top predators in the forest that keep the populations balanced. If you don't have the top predators, when you got all the wolves out from the forest, what happens to the deer population? Okay, well then what happens to the health of the deer? Same thing here. We need the trophic layers so that we get population control and biodiversity in our soil food web, right? So even though the amoebae and the other protists aren't directly symbiotic with the plant roots, they're keeping the ecology in balance. Without them, the ecology will not be able to stay in balance over a long period of time. Okay, there's your ciliates. Okay, and they are going to have little hairs. That's the way you can kind of tell them. I'm going this really fast here. Here's a bacteria-feeding nematode. The reason you can tell is look at its mouth parts. It's hard to see that there. Its mouth parts here have the way we tell one nematode from the other is we usually get its mouth parts. Okay, so that's the mouth part of a bacterial-feeding nematode. Here is a fungal-feeding nematode, and what you have is that this is the nematode, and that's the fungal hyphae, and it's sitting here. You can see it's basically popped on, and it's eating away at that. That's also a good guy. Okay, bacterial feeding and fungal feeding nematodes are good guys because they're the predators on the bacteria and the fungi to keep them under control, right? There's a predatory nematode. Guess what? This is the root nematode. Now, here's the easy thing. Okay, and this thing, what happens is there's this little rod. It's like a doorknob with a rod, and it can take and jam that into the root. That's the bad guy's mouth part. Remember that. All other mouth parts are good guys. Don't worry about it. Okay, so I'm trying to get this as simple as possible, right? Just remember if you see that as a mouth part, that's a bad guy. Everybody else is good. Okay, easy enough. All right. Oh boy, I don't have time to go through that. That is bacteria versus over succession. This was inspired by a diagram that Matt Powers did, and I did my own version, putting a lot more information on it that was very specific and technical. Don't have time for all that, and I don't have to go through the effects of plowing. I will just say that one of the things, then I'll finish up, and then we're going to go into, yep, we're right at it. I got 15 minutes to get onto my the higher you get the leaf bricks, what starts happening is different groups of insects drop out and will no longer attack your plant. If you have aphids on your plants, then your leaf bricks is below the 6 to 8 range. You get it above about 8, the aphids will not even land on the plant anymore. They can't digest it, and they will avoid it. So if you have aphids all over your the leaves of your food crops, it's like that's a sick plant. Okay, from about 7 to 9, the sucking insects go away. 9 to 11, the chewing insects like caterpillars and Japanese beetles go away. And we always know those are some of your hardest ones to get rid of. And finally, up at the 10 to 12 range, the grasshopper group, which includes like the crickets, the katydids, and the locusts, they go away. If you can get your leaf bricks up to 14, a plague of locusts will fly over your field, look at it, and refuse to land. How's that?
Well,
They like,
awesome.
yeah, okay. So this is the right way. If you want pesticides, let the plant make its own pesticide. That's what you do. You actually feed the plant to be that healthy. And when I was learning about this particular thing from Kay Baxter out of New Zealand, she would take any plant in her garden that had a leaf bricks of under 10 and just throw it out. And we look at it and go, that's just beautiful. I mean, way more beautiful than what you'd find in most gardens. She's measuring it going, nah, that's only got a leaf bricks of 8.
You can't feed it at some point and add to its health?
Well, she would basically, the way she was feeding everything, she's like, if it's already not that healthy at this stage of life, there's something wrong with this plant, we're going to get rid of it. Right? Because she's been doing this for years. Right? Now, you're not going to hit 10 to 12 in the first year. Right? What I'm saying is, this is our goal, that after you've been working in soil for a long time and you should see that you're going to start off with a leaf bricks of like 2, okay, or 3. Yeah, and then you're going to see as you work with this and get it better and better, it's like, oh, look, this year I got a leaf bricks of 4. Oh, now up to 6. Now up to 8. And then where Kay is after years of doing this, like her garden is so amazing. It's like, and she has people come there and eat the food and have spontaneous remission of disease. Okay? Like diseases that medical science say are incurable. And so she calls it spontaneous remission because you can't claim medically that that eating the diet cured any kind of disease. So we don't make that claim. What we can say is that maybe if you feed really nutrient-dense food to human beings, that can help their own body's response to disease. I think we can get by with saying that, okay? That's scandalous, yes. All right, but when you get people who have documented, in a documented sense, have remission of 10 years worth of insulin-dependent diabetes after 8 months of eating the food, that's kind of interesting. We'll just say that, okay?
Whose garden is this?
It's Kay Baxter at Kalunga Institute in New Zealand. Yeah, okay. All right, that's what I had. Now, here's what I need to do as we get into the microscopes. Give me 1 second to switch over to this document. And what I'm going to do is I'm going to send this document out. I'm going to give it to Nicole to post. But this is what we're going to be using. I should have printed some of it. But
If only there was a printer here.
maybe we can get a few printed. Okay. All right. So microscope requirements. When you go looking for them, read this. You need, a compound microscope. Here's all the stuff. Make certain that the lenses are parfocal. What it basically means is they all have about the same focal length. What that means is when I switch the objectives, and you switch them by rotating from one to the other, then their focal point is almost the same. That means if you're in focus at a 20x, then you flip it over to 40x, the 40x will be almost in focus. So you're not having to make massive adjustments. It's really fast and easy. So that's called parfocal lenses, right? So what we're looking at here are our eyepieces. Okay, these are our 10x eye. He's gone to putting really camera on top, because if you have to stare through these things for hours, they'll burn out your eyes after a little while. So he actually doesn't almost even use his eyepieces anymore. He has a big 4K monitor, and he has a really high resolution camera, and he's working off of that. And so he's sitting there and he's working on it. He's looking at this 4K monitor, right? And that's actually a really nice workflow. Unfortunately, this monitor is, I believe, running in the 1080p mode because of the way we got things set up. So we're going to see it at a little bit less resolution today, but we'll be able to tell fine, right? But what you're saying, this is called the stage, okay? And the stage is where we're going to put our slide. And then as we get in there and start working with them, we'll show you how to actually operate the controls of the microscope if you've never run them before, right? What you're doing is you're running the focus knobs here, and then there's a stage controls over here. And the stage controls are going to let you move the slide back and forth so you can look at different parts of the slide. And then the focus is going to let you move the stage up and down so that you can focus on the slide, okay? That's basically it. And you're going to have down here, if I hit this little button, I get my illuminator, okay? The illumination comes up to the condenser, and the condenser basically focuses that light onto the slide. And when we come back, we're going to start by how to actually focus the condenser so that the light is focused nicely on the slide, because if you don't do that, you're never going to get really the cleanest image. So we're going to start with that. Then we're going to look at how to prepare the slides, get them onto the stage, and start looking at them, okay? You will notice that until I started demonstrating to you, I had a cover over the microscope. Why is that? Dust. If you are not actively at the microscope using it, put the dust cover on, because you'll be surprised how quickly dust will get onto all your optics. And then you're looking at it going, is that a bacteria or is that a little piece of dust on there, right? And what's going to happen is every couple years, you're going to have to probably get your microscope professionally cleaned, even if you take good care of it, because it's just going to get dirty. And you're going to be like going cross-eyed trying to figure out whether that's something on a slide or whether it's just a piece of dust on your optics.
What's that cost?
It can cost $100 if you, according to where you get it and who's doing it, okay? So here's what I want to do. Okay. Other equipment you're going to need. Let me just hit this really quick. You're going to need several small test tubes. I need them to be graduated like this, so we can actually, what we're going to do is we're going to put like 2 milliliters of compacted soil, and then we're going to add, we're going to go up to 10 milliliters. We're going to add up to 10 milliliters of water in there, and then we're going to actually shake it to get a lot of the life up into the water. Then we're going to use pipettes to get a little of that water and put it on our slide. That's basically what we're going to be doing when we get back, okay? So you're going to need some of these. You're going to need at least 2 pipettes, okay? And you'll see why we'll need 2 in a minute. 2 small containers containing clear, non-chlorinated water. The first is a clean water source for using preparing the samples. The second for rinsing and cleaning your sample pipettes, right? So one's our clean water source. You need to put it somewhere where you won't use it for anything other than clean water source. Because like when we start to dilute this sample, we want clean water. We don't want to have it contaminated by another sample, right? So we have our clean water source. We use that. So we'll need 2 of these, 2 pipettes, several standard microscope slides. We'll look at those when we get back. You're going to need slip covers that are 18 by 18 millimeter size. Because when you look at the numbers I give you on here, the size of the slip cover matters. If you use really big slip covers, it takes your sample and spreads it out over a larger space. And as a result, your counts are going to be different, right? Because every field we look at is a certain amount of space, and we want to dilute everything very precisely. So when you look at a field, you're looking at the same thing every time. So if you're using a 18 by 18 and then later you go to like a much bigger slip cover, that one drop we're going to put on the slide now gets spread out over a lot of space, okay? Yep. And then a sample slide you can use for focusing during the initial setup of the microscope, and a clean lint-free cotton cloth for wiping off. I actually have these little, these are the Chemtex wipes that are designed for being able to dust off. They're low lint. They're designed to be able to like dust off eyepieces and things like that. So when I'm putting things away and I'm taking things apart, I'm cleaning everything. You know, if I get a little dust on an objective, I can clean it and so forth. Don't use normal Kleenex. They have other things on them you don't want. If you're going to use something like this, then get these, something like this, which is a lab grade cleaning piece, okay? That's what you're going to need. And when we get back, let me see, how am I doing? I got a couple more minutes. Good.
How do we collect our samples?
So yes, we're going to collect samples. And what we want to do is, ideally, what you want to do is get us just go down into the soil about 2 inches and get a nice little bit of soil. Now, if we got really complicated, we could do a full sampling protocol. But just for today, go to a place where you know, if we've got something we want to see, and get a good little chunk of soil in a little cup that is not right on the very surface. Because if it's right on the surface, then the sun will like sterilize that and bake it. We want to be down in the soil a little bit, maybe 2 inches or so. We want to get a nice little bit of that and bring it back. And then what we'll do is we'll put 2 milligrams or 2 milliliters of that into this and dilute it, and then off we go. Now the rest of this you'll see, use storage transport, cleaning the slips and side covers, preparing the microscope for use. We're going to go through this when we get back, but what I'd like to do is maybe for the folks that we're going to break here to go to lunch, but if we can have a couple people that will help, we're going to see if we can get the other 3 microscopes upstairs and kind of set and figure out how we can plug them in and like just figure out what equipment we have really fast. And then then over lunch, we're going to try to get this table set up as a slide prep station, okay? When we come back, we're going to bring our soil samples in, and we'll show you how to prepare a soil sample and start looking at it, okay?
How much soil should we get?
Just get maybe a quarter cup. A quarter cup, ideally.
Plastic cups.
Yeah, plastic. Good. Yeah, for this, a quarter cup will be good. Quarter cup, half cup of soil will be fine. Because we only need a couple milliliters of soil. Okay? Any questions before we break?
And we're going to have time to look at that many cups of all these.
What's going to happen is we're going to have hopefully 4 microscopes. And then what we'll do is we'll start creating slide samples, take them to each microscope, and you'll be able to kind of move around. But, you know, when I've done this before, we've taken over the cell microbiome lab of a university, and we've had like 20 microscopes. So everybody has their own microscope to work with or partner up. Today we got 4. We're going to see if we get all 4 working, right?
We know we're pretty confident 3 will work.
Okay, very good. And then I'm going to put the cover back on this. This is one of my personal microscopes.
Yeah, don't mess with me.
Yeah, this is.
I'll be mad.
Yep.
That was more than $400.
But you did die.
Yeah. My really expensive one is my high-end stereo. I have a really expensive stereo microscope that's really amazing.
Guys, so for lunch, Oakland's going to, at some point during lunch, do a burn, which takes about a half hour on the estufa finca. And we're going to get soil samples. We have a couple compost piles we're really interested in looking at. And I know a place where the soil is probably not very soily that might be fun to compare and contrast. Mm-hmm. So I have some plastic cups we can use for that. Lunch is going to go a little longer than it says on the schedule, so we have time to get back here calmly. And then everything will shift forward a bit into the afternoon. So let's go eat. So squeak.
Do y'all want to burn first or burn in an hour?
Burn now.